Cool. So I'm Itzak, coming from the Ben Gurion University in Israel. And first, thank you, Tom, for inviting. Thank you, Christina, for taking care of us. And I'm very, very pleased to be here among friends and hearing wonderful science. And in my group, we essentially involved in trying to understand ecological and evolutionary forces that are kind of structuring and affecting microbial communities in nature in general and specifically in gut environments, meaning their effect of these specific forces on the whole biome in general. And today I'll be talking about plasmids, microbial interactions and adaptation, but all of these are kind of structured under a context of horizontal gene transfer. And maybe we should go back to Seth's figure. You're very popular at this conference, also presented by Thomas. And we all heard a lot about the genome of the host, the genome of the microbiome, either the microbes that are associated with it. Can you actually see the laser pointer? Yeah. Okay. And the microbes that are in proximity to the microbes that are associated with that, with this specific host. And of course, this adds a lot of complexity, but there is an important layer of complexity which is extremely important to this whole, I would call meta-organism, as Thomas would say, or holobiont. And I would like maybe to talk today through this lens. And one of the most important things in embracing complexity, who said to embrace complexity? Was it you? You, Thomas, okay. So one feature or hallmark of being a microbe, specifically bacteria, is horizontal gene transfer, meaning that microbes actually transfer genes from one to the other and between communities. And this is extremely important because they are promiscuous. It's kind of, microbes are kind of letting go to their genomes and shuffling it, and we've shown at 2012 that they can actually cross large phylogenetic barriers when they are inside in one environment. I think there are works by Soren that if you take a broad host plasmid that can transfer genes, it can kind of spread through phylogenies that you've never thought about. And when this actually happens within an environment, it's one thing, but these microbes can actually exchange genes between themselves and microbes in the environment. So essentially, if we think about it, this contributes greatly to genome innovation within a specific ecosystem. If we look at the song and not the singer, the actual functions are changing. You can look at one microbe today, and in 2 months, it won't be the same exact microbe, so you actually need to think about it. And from our point of view, if you think about gut ecosystem, this gets crazy because they are in very, very high density. It's kind of a brothel, if you want to think about it, because they're in very, very close proximity. They can exchange genes, specifically via plasmids, so maybe in nicer words, the gut is kind of a melting pot for exchanging genes between different microbes. And thinking about this or encapsulating this idea, maybe today I'll be talking about 3 different topics or 3 recent works from our group and trying to look at the holobiont through this lens. So we'll start by talking about horizontal gene transfer and its effect on the holobiont or the host microbiome axis. We'll move forward on the drivers, what actually affects plasmids that can carry these genes in a specific environment, what are the evolutionary forces that can affect that, how is it connected to the ecology of the ecosystem, and finally, we'll dive down into a specific example, going from the top down to the very bottom-up approaches to actually understand the role of plasmids within specific ecological systems. Let us start with the first topic led by this amazing person called Sarah Morais. And we'll be talking about horizontal gene transfer and its ability to shape microbial adaptivity to mammalian gut environments, specifically the ability to utilize polymers, dietary polymers. So what are dietary polymers? Of course, we can talk about how important they are or changing ecosystem, but we know that dietary polymers are extremely important for us when we eat. They increase richness. And an important thing to understand about dietary polymers is that by definition, dietary polymers are polymers that we cannot utilize. We cannot actually degrade them or use them unless we have the right microbes to actually do that. So most of them will be coming from the plant cell wall, like cellulose or hemicellulose. Cellulose is one polymer, hemicellulose is it can be more than 3 polymers, like corn arabinoxylan. These are the specific fibers that exist in the dietary food or plant food that you're actually eating. And if you ask most people studying the human microbiome, they will tell you that humans cannot utilize dietary fibers, specifically cellulose or hemicellulose, while, for example, cows cannot do it by themselves as well, but they depend on microbes. And it's indeed the case. Humans usually would not be able to utilize dietary fibers. But recently, or nevertheless, recently we actually found 3 different species in the human gut that have the ability to utilize cellulose and hemicellulose or plant fibers. These species are carrying a specific a complex of enzymes called cellulosomes. I will not elaborate about that. And they can actually degrade cellulose and utilize cellulose. But the conundrum of why humans cannot do that rose when we actually looked at the abundance of the species and their prevalence across human populations. So here on the y-axis you see the prevalence, how many individuals would have this species within them, and the amount of microbes that you see per group of I don't know, either humans or apes, is the actual abundance in the ecosystem. And we find that in industrialized humans, you would find maybe 4% in each population that would have this species within them, meaning that here maybe 4 of you would be able to utilize the plant fibers and might be actually enjoying that and not suffering from, for example, constipation. And if you go into communities like hunter-gatherers or rural communities at East Africa, where they eat a lot of dietary fibers, you see an increase in prevalence of this species. And most interestingly, when you look at apes or non-human primates, about 40% will have them with very, very high abundance. So this essentially only suggests that maybe the westernized lifestyle kind of pushes these microbes out or have some type of a negative effect on these microbes. And I don't know if it's good or bad for us, but this is more or less the case. Now, another very interesting portion of this study was that these species have distinct host preferences. As I told you, we discovered 3 of them. I will be talking now on 2, and I will introduce you to the 3rd one in a second. So we have Ruminicoccus promesians. It's mostly associated with non-human primates. Ruminicoccus hominesians, we named them like this. Afterwards, I realized that I did a mistake because I have a hard time pronouncing their names. But these 2 are actually associated, this one specifically is associated with humans and apes. And the 2 that we find in humans and apes, they are not found in ruminants or cows. And the reason that I'm stating it, because they are actually close relatives to a very important microbes in cow, which is called Ruminicoccus flavificens, one of the main cellulose degraders in cows. And it was very funny for us not to find them inside cows, because we actually found them because we were looking at genomes of this specific species, and suddenly we found a lot of genes that are very similar in metagenomes of humans. So of course, when you see such a thing, you want to understand how did we get them, where did they come from? And in order to do that, what we usually do is evolutionary analysis, or just phylogenetic trees of all the proteins that they share within their genomes, and we see, or we ask whether the clustering can tell us something about the evolutionary trajectory that they actually experienced. And what we find in most of the proteins inside their genomes is the following. We find that Ruminicoccus hominesians over here is diverging from the Ruminicoccus flavificens, which is the microbe from the cows. We see that it's kind of, it's descended. So essentially what this actually suggests, that the microbes that maybe exist in 4% of you, let's say that you're about 104 people over here, have microbes that we somehow inherited or are descended of the microbes that came from the cows. So the romantic way of thinking about it would be that we might have gotten them during domestication. And another very, very interesting thing was to see that there are some microbes that are found only in humans, as you can see over here in blue, but they are closer to their ancestor inside the cow. So this essentially suggests that these are recent transfers from cows to humans, and they are still undergoing adaptation. And of course, this study, this figure that I'm showing you, this assumes, or it takes into account all proteins that are shared across this species. So it shows that there is some type of adaptation in the protein sequence of the genes that they actually carry. But what we actually wanted to fully understand is, assuming that they have this host association and they are not found in cows, apparently they adapted. And not only, we assumed or we hypothesized that it's not only based on the protein sequences. We wanted to understand how did they adapt to the human gut, and looking at the presence, absence of genes, whether their genome composition has changed as a function of invading the human gut. And in order to do that, we essentially created from each genome a vector of genes, okay, from each one of these species. And at the end of the day, we were left out with a matrix of all the genomes coming from the different species, and then looking at the presence, absence of these genes across the genomes. And we used an unsupervised classifier called PCA that all of you are really familiar with to ask how the genes or the genomes are distributed as a function of the composition. And what we find is the following. We see that all the genomes, each one of these dots is one genome coming from one species. All the genomes are clustered according to their hosts, essentially suggesting that all the microbes that are associated with humans have similar genomes, okay? And all the microbes that are coming from more adapted to non-human primates have similar genomes. And we also have this guy, which is the recent transfer that we named Ruminicoccus ruminescens, because it's more similar to cows, that is somewhere in between, still adapting. So at the end of the day, we understand that it's not only the protein sequence, they somehow have similar genome composition with regard to the host that they adapted to. Then you can ask whether they actually lost genes that existed in their ancestors, and then we get these two different clusters, or did they gain genes from the environment or somewhere in between these two scenarios. And in order to ask this, we applied an analysis that is called verticality, developed by Bill Martin's group, which is, I think, one of the best molecular evolutionists that I've ever encountered. What we actually did with this verticality analysis, we asked, or this analysis essentially asked, how deeply a gene is rooted within a specific phylogeny. Okay? When did this phylogeny actually get this gene? If you have high verticality values, it means that it goes long back into the evolutionary story. And if it's very low, it means that it was recently acquired by horizontal gene transfer. And we applied this analysis on the two types, or we classify the genes into two groups. One would be core genes, meaning genes that are shared across all of the genomes of these microbes. And the other one would be host-specific genes, genes that are shared only within a cluster. Okay? So host-specific genes for the hominiscians would be all the genes that are shared between all the genomes here and not with the other ones. And core genes would be all the genes that are shared across all the genomes. And this is the output of this analysis. So you can see that the core proteins, meaning ones that are shared across all, are always having higher verticality values when compared to host-specific genes, which means that the host-specific genes were acquired most probably by horizontal gene transfer from the surrounding environment of these microbes, which is pretty cool. And of course, we all ask ourselves whether these genes have some relevance to the environment itself. And we did a lot of statistical analysis, which was parallel to this one, and we found a clear connection between the annotation of the fiber-degrading genes and the lifestyle of the host itself. For example, if we look at the GH19 over here, which is a chitinase, it's only found in the genomes of the Ruminicoccus permisians, which is a monkey type of a strain associated with the non-human primates. And you all know that non-human primates usually eat insects, and this is being used in order to utilize the exoskeleton of insects. Another great example was this one, this gene called GH98. It was only found in the human-associated strains. And this specific gene is a hemicellulase that is used to degrade corn arabinoxylan. Corn arabinoxylan is the hemicellulose that is found in monocots like maize, and rice that all of you eat, and most of humanity have been eating for a while now, so it's very prominent portion of the human diet. So these microbes, only them acquire these genes, and this, going back to the scalpel examples of Rachel, this gene specifically allows these microbes to degrade a specific portion of your diet, and they actually acquired it and they maintain it. So you can actually think about scenarios in which you want to enrich for these microbes, you can actually use these specific genes. So I hope that I convinced you that a horizontal gene transfer have, I would say, a transformative effect on the hologenome and the holobiont phenotype, because if you have these microbes that got the specific genes, suddenly you can start eating and utilizing plant polymers, embracing complexity. So now, when we understand the impact of horizontal gene transfer on the holobiont, we can start thinking about how it happens and what are the actual forces that drives these processes. And we know that the facilitators, especially in very dense ecosystems for horizontal gene transfer, are plasmids, because they hop between one cell and the other. Those are selfish elements, but they can also hop in and outside of genomes, where they can actually carry very important ecosystem genes like the ones that you just saw. And from Even talk and Soren's talk yesterday, you understood that there is tremendous inflation of data with regard to plasmids, and we don't fully, I think, understand even basic processes as Soren showed us. Most genes we cannot even annotate. So there is a need in actually understanding what are the forces that are driving horizontal gene transfer and also plasmid dispersal. So in order to answer this question, Alva over here actually asked what drives recombination of plasmids, essentially horizontal gene transfer and dispersal. And in order to do that, she used samples from 3,600 different individuals from different studies, and she assembled plasmids coming from all of these studies with different disease state, and assembled, clustered the plasmids, and we ended up with about 11,000 plasmids. And then we can ask these questions of what makes plasmid disperse across human populations, what drives them, which genes, etc. So in order to ask such questions, you always need a reference point, and the reference point in this case was the neutral theory of biodiversity. Usually we assume neutrality, so no other force actually governing the dispersal of plasmid. This was just in order to identify which plasmids are selected. And in this theory, you assume that the abundance of a specific entity in the gamma pool or the gamma diversity pool will be correlated to its prevalence across the population. In this example, for example, the pink plasmid would be more prevalent here than the green plasmids. So in this case, you would have only in 2 individuals the green plasmids. This wouldn't be the case if you would have selection on the green plasmids. Then there would be a disassociation between their abundance and their prevalence, where you would find them in more individuals than you would expect. We understand this. So then we can create a null model where we assume neutrality, and we indeed did that. What you see over here is the abundance of specific plasmids on the x-axis, and on the y-axis their prevalence across populations. And this is the null model here in orange, where this is how a perfect neutral behavior would look like, where there is a very tight connection between just abundance and prevalence across populations. And each one of these dots are essentially one plasmid. And what you can see over here, we were actually very surprised by it, that most plasmids obey neutrality. They're dispersing in a very, very neutral manner. They are not being selected for. Only in specific cases that you see over here, you have disassociation between what you would expect, the abundance of a specific plasmid, and its prevalence, because you see that these plasmids are more prevalent than what you would expect by their abundance. But at the same time, you have plasmids that exist in 90% of the population that are on the exact neutral fit. So the fact that you see a plasmid all over the place doesn't say that it's being selected. And of course, we wanted to continue looking at the ones that are selected, but as you all probably think, like also from the way that I explained, these plasmids reside within host cells, meaning that they are subjected to the way their hosts actually disperse. So in order to cope with this, what we did is we ignored whole plasmid, and we started looking only on plasmid segments that might carry genes and the way they shuffle. And we asked ourselves whether they also obey neutrality in each one of the disease states. And indeed, we find that almost in all disease states, not almost, in all disease states, they actually correlated very nicely with neutrality. And in each one of the states, we do find genes that are deviating from neutrality. And interestingly, and just as a test case, these genes usually would distill the most important ecosystem genes. They would carry things that are very important for their microbial hosts. For example, you see this orange thing over here. You find it in the healthy individuals, the people with irritable bowel diseases, or we call it glucose-related diseases, but it's kind of diabetic. They are all getting treated, but also the healthy humans have this beta-lactam-resistant genes. All of them have it. And this is like resistant to ampicillin, and it's not very surprising. We're being fed with antibiotics from the moment that we are born. This is just a test case to fully understand that plasmids will distill and pinpoint specific genes that are under selection, and they're the easiest way to actually capture these functions from the environment. And to those of you with sharp eyes, you see that the feed, the R square, how close are the dots to the feet, is lower for the inflammatory bowel disease when compared to the healthy individuals. And this, again, suggests more selection in this disease state, and we wanted to maybe delve deeper, or using a scalpel, deeper into this distribution and the genes that are being selected. And in order to do that, we created a plasmid segment sharing network, disassociating ourselves from the plasmids. So in this analysis, there are no whole plasmids. And we created a network between two individuals, A and B, if they share a plasmid segment between them. So you would have an edge that would be a plasmid segment and two nodes of two individuals. Then we can create this network. We created a null model where we assume randomness. So we're now asking which genes are different from randomness or being selected for, and we can have a p-value on each one of these edges, so we are selecting only the significant ones that are deviating from randomness. And I will just show you the results and explain a bit. So this is what we got. You remember that they are coming from different continents. So each one of these nodes is an individual, and the edges are connections based on the plasmid segments. So as you can see, and these are only the significant ones, the ones that are being selected for. And you can see that somehow healthy individuals are being confined by geography, okay, with their ability to exchange genes, or plasmids do not recombine across geographies. And of course, we can think about scenarios that lifestyle, diets, etc., can confine this transfer of genes between different microbes within geographies. But when we look at the inflammatory bowel disease and glucose-related diseases that are also considered as inflammatory ones, we can see specific or breach of this confinement. So they kind of cross geographies. And this, again, suggests that the selection pressure imposed by the holobiont phenotype, or the ecosystem state, selects for specific genes. And this is indeed the case. We find genes that are highly relevant to the ecosystem themselves. Just as an example, we find aerobactin synthase, which is a biosynthetic enzyme that creates siderophores, allowing specific microbes, in the case of IBD, to actually scavenge for iron, and it's considered one of the markers for IBD. But you see it a lot on the plasmids because, again, they are distilling the most important ecosystem genes when selection is involved. So to summarize this, we can think about plasmids as dispersing and recombining in a neutral manner. Now when I'm saying it, it sounds obvious because nothing is governing recombination or dispersal. It's just they're moving around. But whenever selection kicks in, you can find the recombination or the plasmid being selected for with the most important ecosystem genes. And we've been arguing for a while that if you want to see what's important in the environment, you just need to access the mobile genetic pool. So up until now, we looked from a top-down approach, looking at the ecosystem itself as a whole, but we wanted to drill down to a specific gene on a specific plasmid and to understand how does it affect the ecosystem in a very, let's say, bottom-up approach. So essentially trying to ask how plasmids affect microbial interactions. And again, Sarah over here, she looked for a plasmid that breaches geographies. It's found all over the place, and it was found at about 700 samples of the 3,600 samples that we could look at, and also in 14% of the cows. And those of you that know me know that cows are my favorite research model, and I like them a lot. So, and also this is a very easy model to actually approach and to study. So just to show you which plasmid it is, it's like a plasmid that has a lot of maintenance gene on it, and a specific accessory gene. Accessory gene is something that is not involved with the maintenance. And we found it all over the place. We also annotated this plasmid and the gene into the Enterococcaceae family, which was also very interesting for us because this is a gut resident family. And when looking at this gene, and this is mainly the main reason that we move forward with that, we understood that this specific gene, it's here, it's called 1,3-propanediol dehydrogenase, is part of a metabolic pathway. So we immediately thought about the fact that this plasmid might contribute to the energy balance within the ecosystem itself. But in every plasmid that we looked at, we only found this gene without the overall operon. It's kind of ruined our amazing hypothesis. So we didn't understand the context of this gene and its importance to the environment until we actually read in the Wikipedia that this gene is actually, this compound, 3-hydroxypropanaldehyde, is a toxic compound, antimicrobial compound. And this, of course, raised the hypothesis that this gene might be detoxifying this compound. More importantly, this compound is called reutherine. And I don't know, to those of you that are dealing with gut microbes, you know that Lactobacillus reuteri produces it, a very permanent resident of the gut, previously called Lactobacillus reuteri. Eubacterium hallei, also a permanent resident of the gut, produces it. So we started thinking maybe there is something over here. More importantly, this guy called Professor Jens Walter, or Jens, is a friend that actually works with this microbe because you cannot buy reutherine. So he sent me the microbe, and he also sent a protocol on how to extract reutherine. And what we did with that, we extracted it, and we created a targeted metabolomic approach where we can actually measure reutherine in the environment, asking how relevant it is in the ecology of this environment. So looking at 20 cows, easier than humans, no Helsinki ethics, just sampling, and we found like 50% of the cows having reutherine within their gut. And more importantly, we could actually understand what are the physiological concentrations of this compound. Why? Because Sarah is amazing, and she used these physiological concentrations to isolate the microbes that carry the plasmid, okay, as a selective agent. And lo and behold, we actually found a microbe that has the plasmid within it, Enterococcus faecalis, belonging to Enterococcus family that we predicted that it will actually carry this plasmid. We also verified this by PCR and also by this cool method that we're now about to send, which allows us to actually look at plasmids within their microbial host at the single cell level. We see it distributed all over the place. And here, this is a focal picture of a specific gene within the genomes of these microbes. So now we can have a test and try to understand the ecology of the ecosystem through the plasmid lens. So in order to do that, we ordered an isogenic strain for the DSM that doesn't have the plasmid. We cloned the plasmid into this strain, not an easy task, and we compared it to the isolate that we isolated from the environment itself. And we exposed it to reutherine. So it was very nice to see that both the isolate and the isogenic strain are resistant to reutherine, unlike the one that doesn't have the plasmid, which essentially says that causality means that the plasmid is directly responsible for the resistance to reutherine. Then we said, whoa, we did all of this work just to find defense mechanism against a toxin. And actually, we said, yes, this is what we did. And Sarah said, okay, let's sit and think about this thing. And she has a lot of kids, Sarah. They see a lot of Nemo features. And inspired by this sea anemone and the clownfish, we started thinking maybe there is the toxin enables, and the microbes that actually have this plasmid that fights this toxin, enables these two species to have their own clustered environment where they can exchange metabolites or cross-feed each other exactly like the fish and the sea anemone actually do. Because there is not much sense, we didn't find this plasmid in other families. And in order to test this hypothesis, we actually grew these two microbes together and alone. And in each one of the cases, each one of the microbes grew better when it was next to its counterpart in the presence of the plasmid, okay? Meaning that potentially they cross-feeded each other, and we are looking at a mutualistic relationship here, which is crazy because they're trying to kill each other. And we wanted to see if it actually exists in nature. And when we did that, we actually correlated one microbe to the other. And what you can see over here, it's a log scale, that whenever the plasmid is in the environment, here, you see this change, you can find very tight correlation between the two. So these three entities also correlated in nature, which suggests that they actually maintain this relationship in nature. We went forward with untargeted metabolomics to understand how do they feed each other. And we did that. Apparently, one produces stuff that the other one consumes, and vice versa, by just feeding them with the supernatant of each other. And of course, we bought these metabolites, and we fed them with these metabolites, and this will increase their fitness, essentially suggesting that this plasmid somehow maintains or fosters or creates an interaction between these two species, which is positive. So essentially, it's kind of suggests a new paradigm for thinking about plasmids of higher order interaction. So we have one section when these two, or maybe telling us that life is not all black and white. So it's more complexity to it. So you have two entities that are actually trying to kill each other, but the presence of a defense system in one entity enables these two entities to move to a mutualistic relationship where they can actually cross-feed each other and maintain themselves in a very close environment and to have, I don't know, economical flourishing interaction. So with this, I will just summarize. We started by talking about horizontal gene transfer and its impact on the whole genome. We moved forward into understanding what drives the drivers to understand that it's mostly neutrality, and when selection kicks in, we start seeing specific genes that are being selected. Then we delve really deep into the environment and trying to understand how plasmids affect and maintain ecological systems, and we see that they can actually maintain very positive interactions between species and stabilize them. Otherwise, they will just kill each other. And finally, I will say, thank you to the people that give us the money. Also, the 3D Omics. Anton, thank you. It's not you, but in proxy. My friends, dear friends and collaborators on these works, and new friends that I've met that we hopefully will show this stuff, like Anna over here, and my amazing group of people that I'm working with. I'm just showing you stuff that they're doing and communicating it, but they're actually doing really, really cool stuff. So I really admire them, and that's it. Thank you.
So I think maybe we have time for one quick question before we go to break, if anyone. Okay.
Okay, I can't promise it'll be quick, but
Okay.
wow, so creative, excellent storytelling, always. Thank you, Itzak. My question goes back to the first part of your study where you found the cow lineage that presumably moved to humans. And what I noticed there is that there was a long branch to humans, and there wasn't a lot of diversity in humans, but there was diversity in cows. And to me, that suggests maybe a single founding event or a bottlenecking event that reduced the core genome diversity of that particular human lineage that's now derived from cows. So I wonder what you think about that reduced diversity in a bottleneck or founding event. And then the other part is, where do the horizontally transferred genes come from? So I'm sure you can put some kind of bacterial host onto those, and how diverse was that pool? Thanks.
Cool. We'll just do this. So actually, I was talking to you about this study in Bilbao once. So, so what you see over here is one scenario out of two that we found. I'm just giving you the whole picture, which this was the most prevalent one. We also see co-speciation, meaning when the host speciate, the microbe speciate. But we do see, as you said, diversification within a specific host. It suggests it could be clonal expansion, meaning from one founder species that expanded, or converging, yes, but we cannot preclude one or the other. Also, the co-speciation I didn't show here because I'm not really comfortable in talking about it because it exists in about 20–30% of the overall proteins that we looked at. So essentially, the answer is I do not know, but I would guess on expanding. But as you can see over here, this one, Runicoccus ruminis, is only found in humans, but this is way more similar to the cow species, meaning that there are more strains that are invading now our gut and are kind of adapting to it. So you have, it's not one event, you see continuous events. With regard to this species, you know, it's somewhere, the truth is somewhere out there. And with regard to which microbes specifically for the GH98, I think we identified it within the Clostridia, but you can actually pinpoint different genes. I don't, or the Butyrivibrus, maybe the Butyrivibrus, yes. So you can actually find where they are coming from, yes. But it's kind of, as I said, brothel or melting pot. Yeah.
Okay, so yeah, now it's time for the break. Thanks again to everyone, like all the speakers of the session. So please.